Professor Deshpande.
Thank you very much, Ishan. Thank you, Sumit. It's a great pleasure to be here, and thank you for the embarrassing introduction. So what I'll talk about today is on this sort of mechanics of living matter. You'll see that it is on the mechanics, there is significant mechanics, but it also has lots of other topics that have to be included, essentially because we are discussing a living system. So the motivation really is what Schrödinger, in his absolutely wonderful series of essays in 1944 wrote, when he basically says, the essays were called "What is Life?" And he defined sort of one of the main definitions he gave was, living matter, how it differs from inorganic matter or non-living matter is that it evades thermodynamic decay by maintaining, in a tongue-in-cheek way, he said, negative entropy. So that is the sort of motivation for what I'm going to be talking about today. And I will use a very reductionist approach, typical of engineering. And the question is, can this go anywhere? Then can this tell us anything about some system that is so quintessentially non-equilibrium? So the topics approximately that I'll talk about is sort of some background into interesting phenomena that really motivates this whole thing. What I would call non-thermal fluctuations of a single cell. The whole topic is everything is on a single cell, really. So non-thermal fluctuations. Then to say, can we develop a statistical thermodynamics framework for this non-equilibrium system? Now, you'll say, how come you can talk about statistical thermodynamics and non-equilibrium in the same sentence? But that's the game that we'll try to play. And then, of course, say where we can go. What you can achieve by doing this. So let's just talk about some typical and maybe counterintuitive examples of what happens. And I'll start with relatively old experiments. So if you put cells and you put them on a substrate, so on a very soft substrate, 1 kilopascal gel, stiffness gel, or a glass substrate, you see that the cells spread very little on a soft substrate, but spread a lot on a stiff substrate. And if you then look at what is the projected area of the cell as a function, here it's plotted as a function of collagen density, but for different stiffnesses of substrates, as you can see here. So let's just fix the collagen density and go upwards. So with increasing stiffness of the substrate. What you see is that, of course, that the projected area has increased. And you see these error bars. This is typical in biology. You have to put these big error bars. And what this is basically saying is that the different experiments, what was the variation that you got when you repeated these experiments? And these are much bigger variations than you typically see in normal material science. But the interesting thing is that this variation seems to increase with increasing stiffness of the substrate. And this is a bit bizarre, because if you say that this is just because every cell is different, then why will that variation keep changing with the stiffness of the substrate? So what you can see here is that people have thought about this problem of a spreading of a cell on a substrate, and they will typically think of it as spreading of a liquid droplet. But you can see clearly that this is not like spreading of a liquid droplet, because there are all these variations, stiffness-dependent variations, etc. So it's not just a simple variant of a spreading of a liquid droplet. Similarly, there's something even more bizarre that happens. So any cell, whichever cell you take in the body, has contractile elements. Of course, a muscle cell will have much more contractile elements, and these are active contractile elements. So if you put a cell on a substrate of, effectively just a thin elastic film, you can see the film has wrinkled because the cell has tried to contract. And as it tries to contract, it wrinkles the substrate. And these are the stress fibers, which are these contractile fibers, very much like what exist inside what exists inside a muscle cell, and they are contracting the cell. Now, and the way it functions is that these fibers look something like this. These are these active myosin motors that exist inside these fibers, and these myosin motors walk on these filaments, and that's how a muscle will contract. So that's a very active process. It's using energy. Now, you'll say that if you kill those contractile elements within the cell should spread more, right? Because you're stopping the contraction. And effectively, something totally different happens. So this column is a control column. You just put the cells on a glass substrate, and after 90 minutes, they've spread a lot. But suppose you do something else now, and you basically take this the cells and you add this element called BDM into it. It inhibits contractility. Then what you find is that it's not that the cells are spread more; they're not spread at all. So something very counterintuitive has happened. And these are the sorts of features that we really want to try to understand. Similarly, the shape that the cells take on the substrate is also stiffness dependent. So if you look at what is the aspect ratio, what do you mean by aspect ratio? You put a best fit sort of ellipse around the cells, and you look at what is the sort of the ellipticity of the ellipse, or what is the aspect ratio of the ellipse, the ratio of the major axis to the minor axis. You see that the aspect ratio of the cell increases with increasing stiffness of the substrate, but so again does the variability in the response. And this variability is something that I'm going to keep coming back to because this is very critical. What else happens? Cells are guided by the environment. So suppose you take cells and you put them on fibers like this. What you find is that the cells start aligning in the direction of the fibers. Now, why is this important? This is terribly important because that's exactly how tissues form. This is the key to tissue morphogenesis, basically. And I'm going to come to these experiments in great detail because these are the experiments that we are going to use to really understand various parts. So we did these experiments where you paint fibronectin lines on glass substrates, and and each of these fibronectin lines has a different width, and you seed cells on this substrate. And you ask so these are the experiments. So this is 50 micron width, 160 micron width, etc. And then you basically say, what is the orientation that the cell takes on these fibronectin lines? And the reason for this is we are trying to do a very simple experiment. And how do you know what is the orientation? You draw an ellipse around the cell, and then you draw the major axis of the ellipse, and you find what is the angle of that major axis with respect to the fibronectin line. And what you get is now I'm going to discuss this in much more detail as we go along. We plot what we'd call an order parameter. What do I mean by an order parameter? It's something like a nematic order parameter. You basically look at what is this angle phi, and you plot the average of cos 2 phi square plus sine 2 phi square. Why have we chosen this number? So that if all the cells are ordered in the same direction, then this number will be 1. If all the cells are completely randomly oriented, then this number will be 0. So that's the idea. So what you see is that the ordering of the cell is decreasing with decreasing width of the stripe, this width basically. And that you'll say is obvious, but what's not obvious is that the size of the cell is only 160 microns, but the cells have started ordering with the lines even when the striped width is 600 microns, 800 microns. So somehow or the other, the cells seem to be sort of feeling the edges of the lines, even though the cells are so much smaller than the line itself, basically. So this is, again, one of these very counterintuitive things. So the question is really, this is where we're going to go to. How do we understand some of these phenomena? And the real thing which we have to appreciate here is that the only way you can understand this phenomena is that you have to bring the living aspect into it. Without understanding the living aspect, or at least trying to understand the living aspect, none of this will work. So if you take a cell and put it on a glass substrate and look at it, and this is all in minutes, what you see is that this cell is not stationary, right? This cell is constantly changing shape, it's constantly changing size, it's a highly dynamical system. So you can't think of this as a piece of rubber or a soft, some kind of a simple viscoelastic material or something like that. This is a highly dynamical system, and this is going to be the key to understanding everything, understanding this highly dynamical system. And so if you now do this experiment, typically, how do you do this experiment? You basically take cells and you put lots of them on a glass substrate. And then at some point you come and add some reagent that basically just, freezes the cell in a given location, and then you can stain it for various proteins. What you have done is you've caught this cell in a different state of this fluctuation. So each of these cells is different from each other. And the reason it's different is because you've caught it at a different state of this fluctuation that you had just observed, basically. So now the question is, these fluctuations, which I'm calling it fluctuations because they were not really it was not doing anything particular. is these fluctuations are really important. And these are really important to understand. The observed variabilities are really a result of these fluctuations. So what we want to understand is that while the statistics of the observable, suppose we observe cell shape and we say what is the average cell shape or what is the average protein distribution, that's highly sort of repeatable, but there's a huge variability in it basically. So the question is that if the statistics are observables, can we define some kind of equilibrium to understand these statistics? And we cannot really ignore these fluctuations because these are the mechanisms by which cells explore their environment and things like that, right? So that's really important to understand where are these fluctuations coming from and how can we include them in different models, etc. And these fluctuations, you say, okay, even materials have normal fluctuations in them. There's temperature. Now it's obvious, pretty obvious, that these fluctuations are not thermal fluctuations because these cells are too big, basically, for these fluctuations to be thermal fluctuations. So, for example, in this particular case, these were beautiful experiments done by David White's group many years ago. Is that they try to measure what is happening to a single filament, which is in this particular case a filament called the microtubule inside the cell. And so microtubules, as you can see, are fluctuating inside the cells like a piece of spaghetti, basically, in boiling water. Now that is a very strange thing to happen because at 310 Kelvin, which is the temperature of the cell, the persistence length of a microtubule is 6 millimeters, much, much longer than the cell, basically. So given that the persistence length is so long, this microtubule should be, for all practical purposes, behave like a rigid rod inside this cell, but it's not. The measured persistence length, based upon the loss of correlation of these shapes, basically, they measured it to be 30 microns. And now if you put that into the persistence length formula, basically, which is saying, with the temperature, basically you back out an effective temperature of 60,000 Kelvin. Now that's obviously absurd, which is basically telling you that that this is a non-thermal fluctuation that is happening inside the cell. And that's really the, so each filament, every element inside the cell is undergoing this non-thermal fluctuation, which then sort of propagates into cell level non-thermal fluctuations. So you can't really account for all this by conventional statistical mechanics. So what is the source of these fluctuations? The real thing to understand is that if you take this cell, what is the cell? It's, I can't really just look at the cell on its own. I have to look at it in this thing called a nutrient bath. That's the only way to keep it alive. This cell is exchanging glucose and all sorts of other elements with this nutrient bath. So it's an open system that's exchanging species in a very complex manner with its surroundings. Now, these nutrients provide fuel to the cell and contribute to the production of ATP and other sorts of things inside the cell. And it's the hydrolysis of ATP to ADP that's really driving intracellular processes inside the cell, including those cytoskeletal dynamics like the microtubule dynamics that you saw. So some of these dynamics I've said to you, so these are these stress fibers, which are the contractile. And what you can see here is, it's an interesting thing to see here in this cartoon that we are trying to show you here, is that in this cartoon, what I'm trying to show is this, is that ATP comes here, it gives a kick to the myosin motor, and this kick is what is basically resulting in this kind of fluctuations. It's one of the sources of the fluctuation. So these kicks is one big source of the fluctuations. There's lots of other sources of fluctuations because the exchange of nutrients with the cell and with the environment is not precisely controlled and things like that, right? But even though there are all these fluctuations, and now we are looking at, for example, this ATP concentration inside the cell, is that the ATP concentration inside the cell is fluctuating with time, but its average is pretty constant, basically. So, what all the time is happening is that while there are these fluctuations, it's fluctuations about some mean. These are huge fluctuations, but fluctuations about some kind of mean. It's not that as if something is just evolving. So this is suggesting, again, some kind of new type of equilibrium that's happening. So the question is, given all this complexity, how do you propose? Now, there's an obvious way to propose. You say that we know every single kinematic process that is happening, and we propose some kinematic law for a kinetic law for it, and then we'll have some very complex dynamical system, and we come up with the solution. The solution of the problem is the outcome of the solution of the dynamical law. Now that's impractical because typically there are too many of these processes happening. Not only are there too many of these processes happening, is to write something like that, you'll need to use some kind of near equilibrium Ansage reciprocal relations. We don't know what these kinetic laws are, etc. But that is typically what has been done. I was doing that for many years as well. There's another way to do this problem, and this is what my whole talk is going to be focused on, is some kind of coarse graining statistical inference approach. Now, all of you know what I mean by statistical inference, but I'll tell you in 2 minutes what I'm going to use it as. What the idea is the following: is suppose we have a die, and we ask the question, what is the probability of getting number 4 from a 6-sided die? And I give you no further information, then you'll obviously say it's 1/6. But if I tell you, actually, I've thrown the die many, many times, and I found that the average from all those throws came out to be 4, rather than an unbiased die, which would give me 3.5, then what will be the probability of getting number 4? Now, that's a bit of a trickier question. And that's what statistical inference can help us answer. It's basically to answer such a question with making the least claim beyond the available information or data. And this is the basic idea behind statistical mechanics as well. And the key principle is this, so on the verge of it, trivial assumption, is you say that, basically the principle of a priori probability equal a priori probabilities, which basically says nothing in my opinion, but it gives us so much information at the end of it, is that it basically says that if there are n mutually exclusive and exhaustive events, then the probability of any of those events is 1 over n. Now, on the face of it, this sounds as if this is telling you nothing, but actually huge amounts of information can be gleaned from this kind of statement. And that's what we will use. So now we get into understanding what is happening in cells, and this is the approach we use. So let's take a cell in an environment, for example, a cell on that micropattern substrate. This cell will take lots and lots of different shapes, and we will call that the morphological macro state, because it's taking all these different shapes. And each of those shapes is a morphological microstate. Now, within each morphological microstate, for each of those shapes or morphological microstates, you'll have molecular microstates. That means you can get different arrangements of the proteins inside the cell for each of these different shapes. And of course, the ensemble of all those arrangements gives me the molecular macrostate. And of course, there's a molecular microstate, which is typically what this is what is, these two things are typically analyzed by conventional statistical mechanics, is, you have molecular microstates and then this ensemble together to give you a molecular macrostate, which is an ensemble average of those. Now, what is the good thing about what's happening in cell mechanics is that the difference, the rate, the time scale over which the molecular macrostate is fluctuating is on the orders of femtoseconds, nanoseconds, and it's happening over a length scale of nanometers. While on the other hand, what's happening at this higher morphological scale is happening on a length scale of microns, and it's fluctuating really slowly. You saw that, You saw that in those movies of the cell fluctuations. You saw that it was fluctuating on the orders of minutes, basically. So we're going to use that piece of information to basically try to come up with some kind of theoretical framework. So the framework we will do is an entropy maximization idea. And this is an a priori assumption we will make, just like it's made, and we will see how far it goes in agreeing with experiments, really. So we can write the total entropy, which is the entropy of a molecular microstate, in this way, and we can split it up into the joint probability of a molecular microstate and a morphological microstate, right? I and J. And then simply use Bayes' theorem, and we split up the total entropy into this I gamma, which is the entropy of the morphological microstates. That is all the shapes. What is the entropy of all the different shapes it can take? And this IM is the entropy of the molecular microstates. That is the entropy of due to the different arrangements of all the molecules. So we have these two different entropies, But what we are going to do is that we are going to say that because this entropy is evolving on the time scale of nanoseconds, while this entropy is evolving on the time scale of minutes, the evolution of these two entropies is temporally decoupled, and we can maximize these two entropies independently, and that's how we can then maximize the total entropy. If these timescales were coupled, then we are in trouble, because then we'll have to do a joint maximization. And that is what I will not do. I will assume that they are decoupled based upon the experimental observations and then proceed. Okay, so this separation of timescales allows us to use entropy maximization at these different scales, at these different time scales and length scales. So let's now take hold of a single molecular or single morphological microstate, which for all practical purposes, but it's not an exact thing to say, is let's us assume that it basically represents the shape of a cell, right? So it's one of these shapes, but it actually means also the internal strain state as well. It's not just the external shape of the cell. So we take one of these shapes and ask what will be all the possible arrangements of molecules inside it. And how would you do that? You'd basically maximize the molecular entropy, and you maximize the molecular entropy subject to constraints. And so for a given morphological shape, given pressure and given energy conservation, we can then maximize entropy. And that will basically result in a Lagrange multiplier, which is the standard thermodynamic temperature that enforces the energy conservation constraint. So this is standard thermodynamics that is happening. And the outcome is that the most probable molecular macrostate that will occur is the one that minimizes the Gibbs free energy for a cell of a given shape. And that is the outcome of this kind of an analysis. Now, that is an obvious statement that I'm making. You now need to inject biology into this. So we need to say, how do we calculate this Gibbs free energy of a given morphological microstate? And for that, we need to inject this biology, and this biology can be injected basically by, now we have to make assumptions on the biology. And I'm going to use the simplest possible biology, keeping in mind that I want something that's living. So I can't think of this as just an elastic system. I will only include the actin, myosin, stress fibers. I will include focal adhesions. These are the adhesions by which the cells stick to their substrate. I'll include passive elasticity of the nucleus and passive elasticity of the cytoplasm, that is the rest of the cell. So we have these 4 features and only those 4 features inside it. You can add more biology as we go along. And so what are these actin, myosin, stress fibers? So these are these fibers that I've been showing. These are these contractile fibers. They consist of these functional units with these active elements inside them. And then these fibers connect to these things called focal adhesions. And that's how these things will finally connect to the substrate. So that is the biology that is there. Now, this is where all the continuum mechanics will come in. I'm not going to go into any great detail about this, but we'll formulate all of this within the continuum formulation. So we will take a cell, we'll define some kind of an RV, do some homogenization. We'll have to say that stress fibers, or these fibers, can exist at any orientation and at some kind of concentration inside these cells. And that is the active component of the cell, and it's in parallel with the passive elasticity of the cell. So that's the main element. Now, there's a lot of biochemistry that goes into these stress fiber models. And the basic idea of the biochemistry is that these stress fibers can exist either as the proteins dissolved inside the cell, or they can exist in a polymerized shape. And there's some kind of transition from the dissolved state to the polymerized state. And this transition is extremely mechanosensitive, because when you add when you allow these stress fibers to have some stress, you basically change their enthalpy or their free energy very substantially. And that's what brings in the enormous mechanosensitivity into these things. These sorts of things have been relatively well understood. So you can see that as you enhance the stresses, you get more polymerization because you've reduced these energies here. And then also the adhesive proteins, these are the adhesive proteins integrants. These are also mechanosensitive. So you have to put this all within some finite strain mechanochemo model. which is sort of phenomenological, of course. But then you need to solve that kind of mechanical equilibrium, chemical equilibrium problem for a given shape of the cell. Now, this will be a full-scale finite element simulation. I will show you lots of those simulations. For a given shape of the cell, you can then find out what will be the distributions of the proteins that will develop inside the cell. So let's say we have solved that one problem. If we solve that one problem, now we have the solution for this particular shape of the cell. But the question is, we know from experiments that these cells take all sorts of different shapes in a given environment. So the question is, what governs the distributions of these shapes, basically, that the cell can assume? And the idea is then we go to the next level of entropy maximization. And we say, now we're going to maximize what is going to give me the distribution of these shapes is going to be, again, to say that I'm going to maximize the entropy of all these different shapes, which is that maximize the entropy of the morphological states, basically. But the question is there any other additional constraints? Remember, previously we had just used energy conservation effectively as our constraint, and that's how the Lagrange multiplier we had got was the thermodynamic temperature. So is there a new constraint? And this is where the key difference is going to happen now, is we're going to bring in a new constraint, which is basically saying that there's some constraint called homeostasis, which is saying that it's a dynamic steady state by which cells maintain themselves at some kind of, at a steady state. That is, we know that our body tries to maintain a particular temperature, etc. Right? So, so we are going to basically say that we're going to maximize the entropies of all these shapes subject to the constraint that while they're fluctuating, they're maintaining a state of homeostasis. I've not yet defined what I mean by this homeostasis, but that is the constraint with which we will maximize this entropy. Okay, so what do you mean by cellular homeostasis in this context? Cellular homeostasis in this context, basically, I'm going to define it, which is the sort of cellular definition of homeostasis, is that the complex biochemical processes that occur inside the cell tend to maintain the various molecular species within the cell at a fixed specific average number independent of the environment. Now, this is critical. So sodium can enter the cell, can exit the cell, all sorts of species can enter and exit the cell. So in time, it's not that the number of glucose molecules inside the cell is going to be constant, but over time, the average number is going to be held constant and independent of the environment. It's like what you're seeing in these experiment measurements here, is that there's lots of fluctuations, but the average is there, but it's fluctuating about an average, basically. Now, without proof, because I've removed all the equations effectively from this talk, is that we are going to say that the way the sort of one of the remarkable things you can prove if you start with this assumption is that if you use that assumption, you can prove that the ensemble average Gibbs free energy over all those different shapes of the cells is going to be constant, and it's going to be that of a cell in suspension. That is the cell which is not adhered to a substrate at all. So a cell just lying, in suspension. So that is a very strong kind of useful result that you can prove basically using the Gibbs-Duhem identities and other things like that. And that gives us a constraint against which to maximize the morphological entropy. And if you do that kind of morphological entropy maximization, you basically will get what is the probability of a given shape or given morphological shape. And it's given by a formula which looks very much like the normal statistical mechanics, except that this distribution constant zeta that we are getting here is no longer the normal thermodynamic temperature, but it's something else, basically. And you are basically going to choose that temperature, which you don't know, such that the average Gibbs free energy over all these morphological states is equal to that of a G0, which is the cell in suspension. And the physical meaning of this distribution parameter is very difficult to understand. Mathematically, it's basically the conjugate of the free energy to the morphological entropy, but we will physically try to understand what that parameter means as we present the results. Okay, so this result actually looks very straightforward, but of course, the problem is that you need to do these gigantic averagings, basically, and that's not trivial to do in this particular context. So the way, and we call this the homeostatic ensemble, because it's an ensemble that maintains homeostasis. And the way we sort of determine, or we sample on this homeostatic ensembles, is use a normal Markov chain Monte Carlo to effectively construct a sample of the desired distribution. Now, this is well known in statistical mechanics, but we can't use those techniques, because in normal statistical mechanics, you know the thermodynamic temperature. Here we don't know the thermodynamic temperature, rather we know what is the constraint. And so we can't use normal Metropolis algorithms in a very easy way, so we use some Bayesian schemes. And I can tell papers where we've discussed all this. But typically what we have to do is we have to construct a Markov chain comprising of about 100 million steps, which means 100 million calculations. And each of those steps requires a solution, a finite element solution, basically, of that chemomechanical problem, which we had said, which is that continuum problem of a cell with all those species, with the focal adhesions, the stress fibers, the cell elasticity, etc. So this is a very gigantic calculation that we have to do, but it can be done in parallel, etc. So it has been done, okay? So now the question is, what can we learn from such a formulation? So we take this cell, and let's go back now. We are going to go to the experiments of cells where we have painted fibronectin stripes. So cells can stick to these fibronectin stripes of different widths, but they can't go outside it. So what do we get? What will be a typical outcome of the simulation? So let's look at what will happen. And now I'm going to show you a movie of a cell on a stripe which is 50 microns wide. And this is a movie. Now, this is not a time lapse, right? This is randomly picked samples from those 100 million steps, basically. Each of this is one of those steps, basically. So this is only very few steps. And I've colored it exactly the way it's colored in experiments, where the greens are for the stress fibers, actin, the pink is the focal adhesions, and the blue is the nucleus. And this will look very much like what happens in experiments. And suppose we do this for a cell on a bigger substrate, on where the stripe width is 160 microns, then you can see something else happening. The cells do rather different things. There's enormous detail in these calculations. While this just looks like a simple movie, we know, exactly what is the distribution of the fibers in a given shape, how they're what is the concentration, what is the angles in different places, etc. So what can you do? So this is the experiment. So these were the experiments that we did of cells on different widths of substrates, and we have stained this for actin, the nucleus, this is the focal adhesion. This will be how the simulations look. Now, this just looks nice because, the biologists sometimes like pictures. So we show these pictures. They tell you very little because, of course, every experiment, we've got 100 million of these shapes, basically. I can show you any shape that you want, basically, which looks very much like these experiments. So that doesn't tell you much in terms of understanding. What you nearly need to do is you now need to come up with metrics by which you can measure statistics. So, for example, one statistics that is very interesting is what is the shape of the cell, what is the angle that the cell takes on a particular line, etc. So we draw ellipses around the experiments to define the shape. So that ellipticity of the ellipse tells us and this orientation will tell us the orientation. And we can do exactly the same in the experiments, right? Now you'll say to me, why have you chosen to draw an ellipse? Because your cells don't look like ellipses at all. Well, we'll do exactly in the simulations what we can do in experiments, and these experimentalists typically like to define the shape by drawing an ellipse, right? So that's what we are going to do. Okay, so now we can do some statistical quantification. So we can then plot what is the probability, or what is the distribution of the cell orientation for cells on different stripe widths, so 50 microns to very large, 1 millimeter stripe widths, basically. And what we are plotting here are the box plots. What do I mean by box plot? The bottom of the box plot gives the 25th percentile, the middle of the box plot gives the median, and the top of the box plot gives the 75th percentile of the distribution. And these lines give the outliers. So what you can see is that the simulations are the white boxes, the experiments are the brown boxes, is that you're getting very good agreement of the orientations between the simulations and the experiments. But there's a blue regime here and the green regime, which I'm calling regime 2 and regime 1, and we'll define those regimes as we go along. So this is one metric, which is cell orientation. We can look at cell aspect ratio. What you can see is that the cell aspect ratio hardly changes until you reach the end of regime 1, and then it starts to rise like that. Now, this is interesting because while you can see that the orientation is changing, in regime 1, the cell aspect ratio is not changing, but the cell aspect ratio starts to change in regime 2, where the orientation also is changing. But if you look at what is the cell area doesn't really change much at all across regimes 1 and 2. But on the whole, there's some things, of course, which don't agree between experiments and simulations, but on the whole, you get reasonable agreement between simulations and experiments. Now, I'm going to argue that this regime 1 is a pure entropic regime, and we will show you why, while regime 2 is a mixture of biochemically mediated behavior and some kind of entropic effects happening there as well. Okay, so we've shown this picture right at the front, which is looking at the order parameter. It's a nematic order parameter. So these solid line is the experiments, these dashed lines are the simulations, and they're agreeing remarkably well. And so the question really is, what is happening? Why is it that the cells can start to detect the edge of the line, even though the cell size is only 160 microns? Why does it start to order with these stripes when the stripe width is 600 microns, basically? How come it can actually see the edge when this is happening? You know, what's happening really? So that's the thing that we want to understand, and we'll build to that understanding. But before we go to that understanding, let's try to say, why am I calling this regime 2 and regime 1? What is it that is really happening? Well, the simulations tell us lots of things. We can look at, we have a whole thermodynamical framework now, so we can calculate what are the different thermodynamical forces. We can define a biochemical guiding force, we can define an entropic force, and we can define a total guiding force. So this line here is showing the total guidance force as a function of the stripe width. It increases as you decrease the stripe width. The entropic guidance force is these lines here, and then it joins the total guidance force, while the biochemical guidance force is drawn here like that, and it basically goes to 0 beyond this regime 2, basically. And that is why we call this regime 2 to be the biochemically mediated regime, and this regime to be the entropic regime, because the only thing that is happening in this regime 1, the morphology of the cell is not changing, the biochemistry of the cell is not changing, but that morphological entropic force is what is guiding the cell in this regime. And that is why we're calling that a pure entropic regime. It is extremely important to understand that these are not real forces that you can basically say that you can measure, because these are non-Newtonian forces, because this so-called temperature or this distribution parameter is not a thermodynamic temperature. So this is not a Newtonian force. So this is just some kind of a configurational force, if you wanted to think of it that way. There are other forces that are measurable forces that come from the calculation. That is, what is the tractions that the cell exerts on the substrate. And this is which are measured by traction force microscopy and things like that. And so this is showing what would be the traction, the measured tractions on the substrate. They don't change with the stripe width, but they increase dramatically in regime 2. They remain constant in regime 1. And similarly, if I look at what is this so-called temperature, which I had no physical feel for until now, it's showing that the temperature is remaining constant in regime 1, but it increases dramatically in regime 2. Now, remember, in regime 1, we had said that the cell biochemistry is remaining unchanged, irrespective of the stripe width. So in some ways, I like to think of this temperature as giving some kind of coarse-grained parameterization of what is the biochemical state of the cell, really. So that's basically giving us what these regimes 1 and 2 mean. So now the question is, what is the source of ordering? And this ordering, you can say in regime 2, I understand the source of ordering. The biochemistry is changing of the cell, so that's what's changing the ordering of the cell. The question is, what is happening in this regime 1 where nothing, no biochemistry is changing? What possibly could be ordering the cell in this regime 1 when the width is so large and the cell is so small, really? So to understand the source of ordering, I'm looking at the simulations again, and I'm only looking at 2 parameters. There's lots of parameters of the cell. I'm going to look at what is the centroid of the cell and what is the angle, what is the orientation of the cell. And we are plotting the cell orientation on this axis and the centroidal position on this axis for a substrate, for a stripe of width 160 microns. And what does it mean? Suppose the cell is at the centroid. it's basically saying it can take lots of different configurations at different orientations. It can go from -90 to +90. So nearly all orientations are possible. Of course, the 0-degree orientation is much more probable, but there is a finite probability to get even a completely disoriented cell in the middle of the substrate. But if I go to the edge of the of that stripe, basically, you see that really speaking, no orientation other than the 0-degree orientation is possible. And that's the key to this whole game, is that the cells that are at the edge of the stripe can't rotate, basically. And that is why these cells are extremely ordered. Cells that exist at the edge of the substrate are very ordered. And that's what's going to give us this orientational, this kind of bias towards being oriented. And the same thing is the less, and of course, but you can see this boundary layer that's developing. If you make the stripe width much bigger, this boundary layer becomes much smaller, and this effect becomes much smaller. Now, this can be understood by a very simple nematic ordering kind of idea. So if I take a rod and I put the rod on a stripe of width V, okay, this rod has length L and the stripe has width V, width W, basically. And I can develop very simple models now for the orientational order of this rod, basically, in a very simplistic way. So what do I do? So I develop two simple models. One model I basically say is that I take the cell and I fix it in the center, but it can take all possible orientations. And I don't let it translate at all. And in the other model, I say that it can not only rotate, but it can also translate within this width. So what happens now? So if I do that and I say, what is the order parameter? If I've translationally constrained, that is, I've said that the cell can only rotate like that, then if it's in the middle of the substrate, then and the length of this line is smaller than W, then of course it can take all possible orientations, and I'm going to get zero ordering. But the minute the length becomes slightly bigger than the order than W, then it starts to order because it can't basically rotate inside that width, and that's what I'm getting more and more ordering as the width becomes smaller. But now I take the same case and I basically allow the cell to this rod to translate in this manner here. Now, if I allow it to translate, I'm now starting to get ordering much earlier. And that's exactly what is happening, is that the cell can not only rotate, but the cell can also translate along the width. And that's what's allowing this ordering to happen. And why is that the case? So if I look at the entropy, this is the entropy when the translational motion was constrained. If I now, and there's only orientational entropy at this point. If I now say to you that I take the full model where the cell is allowed to translate and rotate, the orientational entropy has decreased. And that's exactly what you see here, is that the ordering has increased because the orientational entropy has decreased. But I'm doing entropy maximization. So why would the cell or the simulation ever tell us that I'm going to decrease the entropy? Well, it doesn't really decrease the entropy, because what the cell has done is that it has traded off a small loss of orientational entropy for a huge gain in translational entropy. And that is what has happened, is that it has gained, total entropy has increased at the cost of slightly reducing the orientational entropy. And that's what's resulting in the ordering that we're observing. It's because the observable that we are observing is only the orientational thing, orientation that we are observing. And this is, the experiments look something like this, which look very similar to this very simple model. So the question is, why did I do all this complication when such a simple model actually worked? Well, the reason I did such a complicated model is because we want to get much more biological observations. So suppose I want to know what is the orientational ordering of all the fibers inside the cell, which is really the biochemistry. And now I've gone and measured all the the probability distribution of the orientation of all the fibers. And now you can see this, the simulation experiment, we're getting extremely good agreement. And that's really what the biologists are interested in, is understanding how the different elements inside the cell are distributed. Okay, so I'm sort of, I'm overrunning time. So this same framework can be used for lots and lots of different problems. And we have done this for lots of different cases. guidance occurs not just by painting fibronectin lines, but by controlling the elasticity of a cell, the substrate. And so if you basically take a cell substrate with a stiff substrate on one side, soft substrate on the other side, you see that the cells will orient themselves on the edge of the, where the transition takes place, and they'll align themselves with that boundary, basically. So the cells will get aligned with that boundary. And so here we show that if we basically, as you saw in these here, is that the cells are orienting themselves along this boundary here like that. That's what we are showing, is that the probability density of the orientation of the cells is that it peaks at 0 degrees when it cells on the stiff side. It will actually be the other way around for the cells on the soft side, but that's a detail that we can talk about later. Okay, so there's lots of different guidance. There's lots of different, this is of course an example of the coupling between the substrate elasticity and the rest of the cell biochemistry. So I've been skirting around the issue of motility because I've been saying that the cells move, etc. But there was no real example of motility that I was showing here. But we can extend this formulation now to something which is truly non-thermodynamical in terms of understanding the time evolution. And how can I do that? Well, I've got myself an ensemble, which I've called this homeostatic ensemble. Given an ensemble, I can then write any kind of dynamics, like example Langevin dynamics on that ensemble. And for example, we take the cell now, we can develop Langevin dynamics. And now I'm going to show you a prediction of real time evolution. So we take the cell from suspension, we put it on the substrate, and then ask the question, what happens? So at this time t is equal to 0, there are no focal adhesions. The cell is a sphere, basically, and it looks something like that. And as you then, now this is a real time evolution. What you can see here is that the cell is evolving, the focal adhesions are evolving. Well, I've not put any time evolution law for the evolution of the focal adhesions or anything like that, but the focal adhesions start evolving. They start evolving mainly at the edges of the cell as observed in the experiments, etc. And then I can now look at how would be the time. So this is now saying what is the time evolution of area with time, what is the time evolution of aspect ratio with time. And you have to do these simulations just like you do the experiments many, many times to understand, to get some kind of sensible averaging result. Otherwise, you just have lots of scatter. So without any new additions, basically, here you can get these kinds of things, that these time evolutions come out from just writing a Langevin dynamics. But what's more interesting is that you'll say, okay, that's sort of obvious. But you can get all sorts of interesting features. For these are experiments for cells on different substrates. What did you see here? Sorry, I should go back one step. Is that the time evolution area sort of reached, steady state in these non-dimensional times of approximately 100. But the aspect ratio is still evolving at 100. It takes much, much longer. So what the simulations have told us is that the area, the observables of the area, the cell reaches its area, but it takes longer to elongate itself and get to its steady state aspect ratio, because that's taking longer. And that's pretty much what is observed in experiments as well, is that you can see here is that the cell area has basically reached steady state maybe in 200 minutes, but the aspect ratio, which they're plotting here as the inverse of aspect ratio really, is evolving, and it's even after 20 hours that the cell has not yet reached its steady state aspect ratio. By the way, these different red lines is basically different experiments, and it just shows you the level of variability that you will get with each different experiment. So there's lots more that can be done in this direction. Where is all this going? We are doing this, so the key is to add more biology. We added only stress fibers, focal adhesions, and some cytoplasm elasticity, substrate elasticity. The question is, cells can detect curvature. You basically can't detect curvature unless you add microtubules into the biology. So that's the game that we are playing, is that you have to add more biology into the models of the cell to basically capture the features. But that's the advantage of having these kinds of modeling approaches, because what it tells you is that what is the minimum amount, or what is the critical element that is needed to capture the physical observation that you're making. So it tells you what does what inside the cell. So, for example, if you wanted to capture how the cells are behaving on curved substrates, we're adding microtubules. We are also taking this whole thing forward, because from single cells to homogenization, to tissue scales, where we are constructing sort of what you could call micro tissues, and understanding what is evolved, how does the microstructure of a tissue, i.e. what is the distribution of cells that can happen inside a tissue at different positions vary, how does the cell density vary, etc. And the idea here, of course, is that the same idea holds true, is that homeostasis happens at the cell scale, it happens at the tissue scale as well. So the same statistical homogenization idea that we use for single cells, we are planning to take forward to single tissues as well, or very simple single tissues. So I should end by basically acknowledging the many people who worked on this. Now, I've not shown all their work, but at least parts of it. Nearly everything I presented was the work of one of my PhD students, who's now a postdoc at the Cold Spring Harbor Labs. Is that this was nearly everything was her work, but there have been lots of people over many years from various people involved in these different projects, basically. Thank you very much. Hello.
Okay, thank you very much for an excellent and timely talk, because I guess mechanobiology is really coming up all over the world now.
Yeah.
What I'm going to do now is, I'm sorry we can't clap for you across Zoom. I tried, didn't really work out too well. There are several questions in the Q&A. What I will do is I'll read out a few of them, and then I will invite the attendees to raise their hands and ask questions directly. You have some time right now?
Sure, absolutely.
Okay. All right. So let me start. I'll start from the first question by Shiv Surendra Chandran, who asks how the persistence length of 6 millimeter was deduced for the microtubules.
Okay. So persistence length, as we know, is basically a ratio of the bending stiffness, i.e. EI, of an element to the KT. So the EI, or the bending stiffness, has been independently measured of microtubules outside a cell. And that's how you would, and then you know that the temperature of the cells is 310 Kelvin, and that's how 6 millimeters was deduced, basically, from the independently measured bending stiffness. Now, as I said, that when you measure the persistence length by a loss of decorrelation inside the cell, it comes out to be 30 microns, which is a very different story.
Okay, here's another question from KV and Surendra, who asks, what are the active and passive elasticity components? Are they related to purely elastic and elastoplastic material behavior?
Okay, no. So I didn't explain all this, and I apologize, because there was no way for me to go into the details of the biochemical model. The elasticity is very simple. It's just simply sort of, what you could call Ogden elasticity, for lack of a better word. So it's just a nonlinear elastic model. The active components are not plastic at all. They're active, i.e., they will violate all of Drucker's postulates, etc. Because what they will do is that it's like putting an actuator inside the cell, and those active models will be basically have parameters like the concentration of the stress fibers, the orientation of the stress fibers. Then they will have elements such as what is the stress that the stress fiber exerts. And they will have, but we don't know what is the concentration of the stress fibers. We don't know what is the orientation. And we use free energy minimization, basically, to tell us what will be the orientation and concentration for a given shape of the cell.
Okay, moving on to another question by Vagish Baghel, who asks how you calculate strain for stress fibers in your biomechanical.
Okay, correct. Sorry, again, this comes back to strain is not defined for stress fibers, because of course they're remodeling constantly. So there is no reference state. The elasticity of the cell has a reference state, and so strain has a physical meaning to it. The whole point is that the stress fibers have no reference state because they dissolve and reform. So you can't really define a strain for them. And hence you have to basically use these kinds of chemo ideas, basically, to understand what is their state. For people who are,
another question from Prabhat.
I know that this is all just words because I didn't put those detailed equations, but I can send papers to people who are interested in these detailed equations.
Prabhat Agnihotri asks whether excluded volume effects in stress fibers influence the self morphology.
Yeah, so now this is a more complicated question, and it's a very, there is no question that excluded volume effects are important. Now, because I have used a continuum model, the only excluded volume effect will be the sort of normal entropy idea that basically, which comes in through concentration, that the entropy decreases as you increase the concentration. Now, that is not totally correct in stress fibers, because these are, to think of a stress fiber as being local at a given point in the cell is not a very good approximation. And it would be much better to think of them as discrete quantities. But, I haven't done that yet. Let's put it that way. That is a bad approximation that exists in the models at the moment.
Kemal Basaran has a question, but he has raised his hand, so I'll allow him to talk. Please go ahead, Kemal.
Oh, okay, thank you very much. Well, I am Jamal Basaran from University at Buffalo. Well, first of all, thank you very much for your amazing presentation, Professor Deshpande. Now, I understand, so the cell is an open system, but if we assume for a limited nutrient, we create a hypothetical closed system. Now, we can use the unified mechanics theory to predict lifetime of a cell. But if we know all the thermodynamic entropy generation mechanisms, so my question is, do we know all biological, chemical, mechanical, thermal, electrical entropy generation mechanisms in a cell?
No, we don't. That is the precise problem, basically. And that is why I did it in terms of the only assumption. If you knew that, then you could develop something much more. If you knew all these pathways, right, then you could definitely start thinking about doing Onsager or something much more of that sort, basically. The problem is, because we don't know it, I've done this enormous coarse graining and used basically something as coarse grained as homeostasis, basically. So partly we don't know it, partly there are too many of these processes that are happening inside the cell, is what I will say, for us to say that we would want to model all of them.
Oh, I see. Okay, okay. Because
it's like saying to me that, sorry, I just say, it's like saying that, of course, a correct way to model an ideal gas would be to model all the molecules. But, in some ways, why do you care, right? You could ask that as a question if you're only interested in the ensemble average behavior.
Oh, I see. Because using unified mechanics theory, now we can predict lifetime of a steel bar, but we know all the mechanisms that contribute to entropy generation.
No,
that's if we don't know the mechanism, then that is difficult. Okay, thank you very much. I don't want
that is precisely in a chemical factory. You would use open system ideas, basically. And so I'll just answer this a bit more carefully. Is that firstly, the number of processes, even if it's large, is very limited in any kind of normal engineering system that we look at. And the second thing is that most of these are near equilibrium systems. So, you have to use the Onsager reciprocal relations to understand the couplings between the various physics. And typically we use Onsager reciprocal relations. The cell is so far away from equilibrium that Onsager assumptions, are known to fail. I mean, for example, there have been very nice recent measurements to show that the detailed balance, that is the underlying assumption behind all these Onsager relationships, actually fails. Experimentally, they have shown that it fails, basically, in living cells.
Oh, I see. Because essentially what you're saying is, like, in unified mechanics theory, you don't use the Newtonian mechanics because the derivative of displacement with respect to entropy is not zero, as in continuum mechanics. But what you are saying is we couldn't be able to calculate that because we don't know all these mechanisms.
That's right. That's right.
I see. Okay. Okay. Thank you very much. I really enjoyed your presentation. It was really amazing.
Thanks.
Okay. Thank you. Shailendra Joshi had a question, so I'm allowing him to talk. Please go ahead.
Hello, can you hear me?
Yes, I can hear you.
Hi, Vikram. Awesome talk. I enjoyed every bit of it.
Thank you.
And you know, it's a lot to digest, so I'm going to read your papers more carefully. But amongst other questions, the one question that I had was in your model that you have constructed, the chemomechano model. I'm sure there must be some kind of a length scale embedded in it, because the fact that you are able to distinguish between the relative distances between the small strip and the big strip and the way cell responds to it, where does the length scale come into picture?
No, the length scale is not inside. So that's a good point, Shailendra. So the length scale is not in for this orientational guidance. The only length scale that exists, and let me find, is somewhere like this so-called W over L, which is basically saying the ratio of the width of the cell or the size of the cell to the size of the strip, basically. So that length scale is present, right? So when I show this kind of a plot, this is the striped width, but I start off with the size of a cell in suspension, which is 160 microns approximately.
Okay. So
that length scale exists, but inside the cell, there is no length scale. In my current model, that's a very bad assumption. It came back to the previous question on the excluded volume, is that it is not, it's absolutely true that when I look at these cells, you can see, right? Look at these discrete fibers, right?
Right.
So there's absolutely no doubt about it from a continuum perspective that you need a non-local model for this, right?
Sure.
But
if I may ask one more quick question. So when you think of, again, the details may be in the model itself, I need to read it, but when you consider the interaction between different fibers in a continuum sense, do you account for the possibilities that these actin binding proteins, they can dissociate and reassociate?
Absolutely.
Okay.
Without that, you can't get anything. So, for example, here you can see that, we've got these actin and myosin, and, there's a particular concentration. So if I go back, so this is where the entire chemistry comes in, is how many of these proteins actually polymerize to give you a fiber.
Okay.
And so you have to take into account this kind of a chemical balance. And that is inherent in all these models, basically.
Great. Thank you so much.
Would you be willing to take 4 or 5 more questions?
Sure. No problem at all. I'll be happy.
All right. Just 1 second. So Babak Ravji, I hope I got his name right, asks, in the absence of external stimuli, can you elaborate on why the cell never reaches an equilibrium state? How in the model can you do you change morphology constantly?
Okay, so I'll answer the second question before I answer the first one, because the first one is a bit trickier. Now, I don't actually make it change the morphology constantly. In the bulk of the presentation, I did what I called a statistical model. So we basically have found out the distribution of shapes or morphologies that maximize entropy, basically. And I'm not telling you anything about time in that formulation at all. So I've just told you what is the distribution of states that the cell will assume. Now, when I do something like, when I go to time evolution, which is something like this, then of course, all that I'm doing is I'm solving this kind of over-damped Langevin equation to come up with these shapes, basically. And that's how I basically, these damped, this noise term, which is that non-thermal noise in the Langevin dynamics, is basically telling you why the cell shape is changing all the time. Now, the first question is, why is it that the cells never reach an equilibrium? Well, in some ways, that's a trickier question, because that's asking, why is life what it is? But I will say that the cells are a driven system, right? The system is just the cell. It's constantly exchanging nutrients with the environment, and we are only looking at the cell. And so as it's constantly exchanging nutrients, that exchange is not well controlled. Sometimes glucose molecules enters the cell, sometimes it doesn't enter the cell. And that's why this system never reaches what we would traditionally call an equilibrium, but I will claim it reaches a homeostatic equilibrium. So it's a matter of how you define the equilibrium, really.
Okay. Thanks. I got four, 5 more questions. I'll keep plowing through unless you stop me.
No, sure.
So Kalpesh Jaikar had 2 questions, mostly about how you are drawing the ellipse about the cell uniquely, and what if the ellipse turned out to be a circle.
Okay, so now, Kalpesh, you're absolutely correct. And I will go back to there is no unique way to draw this ellipse. They are what you would call best fitting algorithms. So what is being drawn here is what we call a best fit ellipse. So what do I mean by a best fit ellipse? You take, you minimize the mean square, the mean square distance between the ellipse and the real shape of the cell. And that is traditionally what is done. So, that algorithm will give you some kind of an ellipse. There is no guarantee that that is a unique optimization, right? When you do that optimization of minimizing that mean square, but it's one way to measure the morphometrics is all that I'm saying. I'm not at all trying to say that this is mathematically a unique way to do it. And what you've said about the circular cell is exactly correct. Is that if you what I will come up with is that if I have a circle, then I will get a unique answer because my best fit ellipse will be a circle at that point because I'm minimizing that distance. But of course, at that point, the orientation of the cell is undefined. Okay. Because there is no major axis to a circle. I should once, by the way, point out, right? Remember that we are doing an entropy maximization. So getting the probability that I will ever sample a configuration that is a pure circle is vanishingly small, given that there are so many other possible configurations, right? It's like saying that, in normal entropy, there is a 0K, i.e. when no molecule is moving. That configuration does exist even at 300 Kelvin, but the probability of that microstate is vanishingly small, basically.
Okay, I should alert the attendees that you can raise hands and ask questions also while I am reading the Q&A in case you have questions. Shiv Surendra Chandran had a couple of follow-up questions. First one was, entropy maximization for equilibrium could be understood for systems where all energetic interactions may be absent. Is that the case for these cells? And the second question was, what is the reason for the initial delay time in simulations before which the cell area seems absolutely constant?
Okay, so let's answer the first question first. I'm not sure I understand the question. So maybe if the attendee could ask.
I am allowing you to talk if you would like to ask a question directly. Please go ahead.
This is Shivasendra Chandran here. So the first question that I was focusing towards is like when you initially started trying to understand the system from the entropy maximization perspective, I was just wondering why the interaction between cells or the interaction between cell and the substrates were not taken into account.
Oh, no, sorry. The cell and so I should have been more clear. Is that the interaction, the system that I have consists of the cell plus the substrate, but not the nutrient reservoir, okay? So the interaction between the cell and the substrate is absolutely taken into account in the entropy maximization because the system that I'm maximizing the entropy of is the cell plus the substrate.
Okay, so what would happen with the cell-cell interactions? Was that?
Oh, so this is every simulation and experiment that I've shown is basically for a single cell. Now, I agree with you that the way the experiment has been done is not a single cell, but the seeding density of the cells on the substrate was so low that the cells would never interact with each other. So this is all for single cells.
Okay, thank you. So that was not clear when I was listening.
Sorry, yeah. Yeah.
Thank you.
Okay, now your second question, which I've forgotten now. What was the
So I was speaking about your the time evolution of the cell area that you are showing towards.
Okay, yes. So the reason why the cell area takes time to evolve is because you start with the cell which is in its suspended state, and then you're moving to we know what is the equilibrium or the homeostatic distribution of the area on a particular substrate. It is moving down because, at least in the Langevin simulations, I can tell you what's happening. In the Langevin simulation, this first term here, can you see my arrow?
Yes.
This is basically driving the system down a particular free energy gradient. And what's really happening is that the time is associated with the time it takes to drive itself down that free energy gradient, associated with the dissipative terms, right? In the Langevin thing, you have dissipative terms, which is setting the time scale, why it's taking time to go down that free energy gradient.
This is some kind of an intrinsic time scale, which
intrinsic time scale, or you could call it, in normal mechanics, you think of it like a Stokes drag. Basically, you say that there's a viscosity in the system, there's a dissipation in the system, which is basically which is adding that intrinsic time scale, or which is associated with dissipation.
Okay, thank you. That was clear.
Last two questions. The first one is from KV and Surendra, who asks whether all the cells in your experiments, are they all live cells, or do you have dead cells in there too?
No. All the cells that have been that are shown here. For example, let's go here. Right. So these would be these are all live cells before they were stained. In the photograph, by the time they reach the photograph, they're dead. So let me explain to you how the experiment is done. You seed the cells on these striped substrates. You wait for 48 hours until they achieve their homeostatic equilibrium. And then you stain the cells to be able to observe these proteins. And when you stain them, they also get fixed in their shape. And so at this point, they are dead, but they were alive for 48 hours until they reach this state. Does that make sense?
Yep, I think that was clear. Nirmal Kumar, I am going to allow you to talk. Please ask your question directly.
Hello, sir. I'm just asking, and not asking, actually, I need your suggestion. If I go to solve a problem, cell interaction or contact adhesion problem, right?
Yeah.
So considering ellipsoidal shape will be more general approach rather than a spherical shape.
Yeah, I mean, the minimum, we will say that, if you want to look at cell-cell interactions, you'll need to have more than ellipsoidal, but minimum you'll have to have is ellipsoidal, as I would say.
Instead of considering spherical, you are suggesting I should go for ellipsoidal, right?
Correct. But what I will say to you is that if you can see that the shapes that these cells take is not ellipsoidal or spherical at all, basically. Mm-hmm. And this becomes even a more severe issue once you get cell-cell interactions, basically. Mm-hmm. So I'm not even convinced that an ellipse is a sufficient parametrization of mm-hmm. cells when you go to cell-cell interactions. But it will be better than a sphere for sure.
Okay, sir. Thank you, sir.
The last question of the day is from Vagish Baghel, who asks if the interaction between different elements, whether it is important here such as in between nucleus and stress fibers. I mean, yeah, I don't know if that makes sense.
Okay, so I can see the question. So the interaction between the different elements is terribly important. So for example, if I keep the stress fiber properties to be this fixed, and I only increase the elasticity, I make the cells stiffer, for example, I will kill most of this behavior. So it's extremely important that the way the cell behaves is its balance. And Dennis Disher understood this 10 years ago, 15 years ago, is that there is a balance between the energy between the elastic energy of the cell and its chemical energy, basically. And if you change that balance by changing the elastic properties, you dramatically change the behavior. And in fact, that's what happens when you change cell type, for example. You've asked the second question, which is what makes a cell active. It's basically the only active element inside this. The only two active elements are the integrins, which is the focal adhesions, and the stress fibers. But primarily, it is the stress fibers. Later on, when I was talking about the curved substrates, I said the microtubules are active as well. But in the bulk of the talk, effectively, you can think the only active element are the stress fibers.
Great. I don't think I have any more questions left. There are no hands raised, no Q&As. So once I dismiss this, we have a clean slate. Let's thank Professor Deshpande once more at our home. Give him a little clap. And Sumit, would you like to wind up the?
Yeah, thanks a lot, Vikram, for this wonderful talk. I just have one announcement. The next talk is on in a week's time, 7th October, I think it is. Konstantin Volok from Technion is going to talk on fracture of soft materials. So hope to see you all there.
And thank you very much, Sumit and Ishant. It was a great pleasure for me.
Yeah, thanks a lot. Thanks a lot.
Thank you. Bye.
Yeah. So we close the meeting.
Thank you. Bye.
Bye. Soft material. So